So good morning, everyone. In this lecture, we are going to talk about cell type prediction. And I should, in the beginning, mention that most of the slides are coming from the latest lectures by Ahmed Mahfouz from Netherlands and also Osa. So most of the slides are from there, and then I added some more slides at the end. So, okay, now it worked, right? So first we need to define what cell identity is. We can define it by these three pillars in this photo that you will see. The main pillar here is, which is central for cell identity, is phenotype and function, which is usually the physical, and also defines by the markers. So it will tell us about the markers about that, and also the functionality of the cell type. So it's the traditional cell type definition comes in this pillar. We also the second pillar here is lineage, which tells us about the past state of the cell. So we can have a taxonomy from developmental origin, for example, where these cells is coming from. And the third pillar is a state, which tells us about the future of this state, or the phenotypes that the cell type can get if it's under a certain condition, or it's a disease condition or something. Those are states of the cells. They are not different cell types. So we want to make that clear here. What we use here, and I will describe in this lecture, is more on phenotype and function. We talk about transcriptome. So we want to define the cell types. Of course, we go, for example, in some methods, we go into more detail and we extract the states of the cells as well. So just be familiar with the cell identity. The other thing we need to keep in mind is that we need a standardized vocabulary when we want to annotate cell types. So this effort has started in 2021, mostly by Cell Atlas projects, that they started to have a cell ontology for that can be used in different projects. So if they want to integrate different data sets, then the vocabulary is similar, so they don't have to deal with the mismatches between cell types. And it includes 5 important information here that you can see. You can do it here, laser point. That you see in the right part. So the EASE A1 is really defining the cell type. It's a part of an organ and tissue. It's located in a certain space. And its developmental origin, where it's that one, and also its functionality. So we need all these 5 information to be recorded in cell ontology. So that makes things easier later. So it has been like in the latest paper that I was reading about cell type prediction, they are really using this ontology. So if you want to use a reference data, you have to turn it to something like this, and then you will be able to use their method. So the question here is that how can we identify cell populations? So now, so far, you had done like quality control, normalization, and everything. You have done the clustering. You did differential expression analysis. You extracted the markers that you wanted. So we have done everything. And now imagine that you have that matrix of the data. You have cells and the expression of each gene in each cell. What we want to get now is we want to get the labels for these cell types. So we want this population of cell types on the right side. That is what we want to get. One way to do that is that you do a clustering, and then you will map either like the markers that you already know coming from your prior knowledge, that a certain marker should be from a cell is for a certain cell type. So you map it on the cluster and then do the annotation manually. The other way is that you can do it in a data-driven way. You can just the way that, for example, you did with differential expression. You extract the differential expression marker genes for each cluster, and then extract the top significant ones, and then look into them, map it on the cluster, of course, and then look into them, and you see that, okay, if you can annotate them. This is unsupervised cell type identification. That is, of course, time consuming. It is not reproducible. Sometimes, like two people, they can disagree on a certain level. So, I mean, of course, you can agree on main cell types, but if you go to higher resolution cell type annotation with like more subtyping, then people start to disagree. And also, it's subjective. So you need to make a lot of subjective decisions if a certain marker is expressed in a certain cell type, or is it really a marker for that one. If it's covered in like there is overlap with two clusters, are they which one of these cluster you wanted to label for that cell type? So a lot of subjectivity here that makes this problematic. So now the question is, can we do this automatically? So we don't have the issue with like we needed to have someone that has the knowledge about all the cell types that we have in the data and has time to do all of those manual things if you have a very big data with different cell types and you want to do it in a very high resolution way. What I will describe here is just in a simple way, I will describe some basic machine learning methods like that can do this for us. What we really want to define, what we mean with this cell type, we just want to define these boundaries between these cell populations. And that can be either by clustering, which is unsupervised learning. We really don't learn anything from the data. We just try to understand the structure or discover the structure of the data, how many groups of cell populations we have, how much are the distances between these populations. So clustering can define these ones, but if we go more to classifications, which is supervised learning, then we want to define, find the description of these boundaries as well. So we want to know, have a function that can describe this boundary for us. And we also are dependent on the prior information. For example, we need to have a data set that has the labels as well. Then our classifiers can learn from that, the relations between the data and these boundaries, so it can be trained and used for the cell type annotation. Just imagine that you have this matrix of the data. You have cells in a row and like features that are genes in this case here. And you also need to have labels for the cells, for these cells that you have. In this case, we have T cell and B cells, like 4 points here. What you do is that you will look at your data in with a dimensional reduction method, and then you're hoping that these points are gonna get close to each other, and they are closer to each other than the others. And by a classifier, the goal is to define or describe this boundary here. And then when you get a new unknown cell, a new data, you have a profile of expression. You want to see what this cell is. So if this cell at this point is just drop here, and you know the function that describes this boundary, then you can easily say that this is a T cell. So that's simply what we want the classifier to do. So now the question is that how we can train these classifiers. So imagine that we have just two-dimensional data here. We have blue and red populations of the data points. For each cell in the data set, we have gene expression of XJ, and we also know the label. We know it's red or it's blue, so we have that one. A classifier is simply a function that will get an expression profile and predict the label for that. So that is simply what it does, and it's just describing the boundary for us. And the goal here when we are training the classifier is to minimize the error. So we don't want this classifier to make mistakes either. So we don't want the blue one to be predicted as red or red to be predicted as blue. So we have a look at this one to minimize that one when you are training. So one of the examples of this the classifiers are k-nearest neighbor. They're called lazy classifiers because there is no actual training there. These classifiers, they keep the whole training data set. When a vector comes, like a vector of expression comes, then they will find, for example, they're projected on the dimensional reduction space, and then they will find the closest example. For example, here it is yellow dot is the closest one is a red one. So then they say that, okay, this is a red data point. We can label it as red. And similarly for this yellow one close to the blue one. And then it will predict if this is a blue or a red. So that is a simple way of describing it. To make this nearest neighbor work, we need 4 things. We need to know, we said that we want to know how close they are together, right? So we need a distance metric. And we need to know how many of the neighbors we should look at. We should look at one of them, or we should look at like 5, 10, 50, how many? It depends on your data, of course. We can also have a weighting function. It's optional, but I will describe it shortly in one slide briefly. And also, we need to also have a way to decide how we want to label this new data, how we want to define it. For example, is it going to be a majority voting, or is it going to be average over these k nearest neighbors that they are close to the data point that we want to classify. So k-nearest neighbor uses Euclidean distance. It is simple, it is quick, and since it's keeping whole data sets, we need something quick to measure this distance quickly, because they need to go through like so many points, depending on your k, of course. How many neighbors to look at? This is what you need to really define yourself. Is it 10, 20, 30? So that is also very important. You should just also keep in mind that this K is used for the whole classifier. If you have unbalanced cell populations and you use like a very high K, so it's going to get very confusing for those small classifiers. So it's very tricky to know what kind of K, which K you need to choose. You can, of course, scan a set of like Ks and then see how your classifier perform based on that. You can have a weighting function, which is unused here in the original format. And to decide to make a decision about which label it should get, it will get the average output among the K nearest neighbors. So here, imagine that we have these 3 data points, 3 groups, like red, blue, and green. If you look at the one nearest neighbor here, you will get all these rough edges here. And when you get, like, for example, a point, I don't know, here, then since it's the closest, you look at only one neighbor, then you say that, okay, this is green. But if you get like 5, look at the 5 nearest neighbor, then in some places you see these white patches that the classifier can't really decide, because all the 5 that are close, they are from different groups, and they couldn't define the boundary there. So it gets confusing. It's a confusing task there. One trick to solve this one is to use weighted nearest neighbor, which called also sometimes kernel regression. The distance metric that is used here is also Euclidean. How many neighbors to look at? We look at all of them. We don't choose any k here. But we have a weighting function that we give the points that are the closest to the data point that we want to classify the highest weight, and those that are further away, the lowest weight. So it's just based on this Gaussian kernel here, for example. So it's the distance. The closest they are, the higher weight. The further away, the lowest weight you get. And for the local point also, to decide which label we want to get, we also get like weighted average. Just to show how it does, just in a like toy, a cartoon way. If you have K1, you get these boundaries. If you have K2, for some of the points, can't really decide because it's close to 2 points that are one blue, one red, for example. So you get a bit of confusing boundaries. But the kernel is kind of solving this by making this boundary smooth. The other classifier that we talk about is support vector machines. They are one of the relatively simple classifiers. And one of the most famous, and of course, and they usually perform very well. So just to describe it, how it does, so we have, just like before, we have these blue and red ones, and we want these describe this decision boundary here. So we can also have this boundary here. That still doesn't make any mistake, right? So it will still be able to divide these two groups without any error. But the question is that which one of them should be chosen is what really SVM is doing. SVM is trying to describe this boundary, like find this boundary and that maximize the margin from both groups of the population here. So if we have this black one, we don't get error, but this boundary is far away from different population of cells. Imagine you have a noise in your data and you want to be away from all of them. But if you have this blue one and you get, for example, a data point here and your reference boundary is reference boundary 2 here, then the classifier will categorize it as blue, label it as blue. But if you remove the boundaries, you will see that, okay, this is closest really to the red ones. So it doesn't make mistake, but it's not correct either. So that's how SVM is doing. So now we want to say that, I mean, the methods that I describe is because I am going to talk about the algorithms that are using these basic classifiers there, so you know at least how they are performing. So now the question is that when we want to automatically identify cell populations, what do we need? We need, using these classifiers, of course, we need training data. We need like a cell atlas that you have information about the cell types, that you need to have the labels there. You need, for some methods, they need prior knowledge. So you need a set of marker genes for those methods. And then when you get the test data, annotated cells, you get a new data set. Then you run it through this classifier and then annotate the cells. And some of the methods will also give you these rejection options. So we'll leave some of the cells that they are not familiar, they weren't trained with, as like unknown cells, which is a very important feature that for the classifier has, and it's a very also tricky task for a classifier to do. They perform differently because they need to make a decision there where to decide that this is unknown based on which similarity, or so we can talk about that one in the coming slides. So in the coming slides, I will talk about this benchmarking paper that was done in Ahmed's group. It's a very thorough way of looking at the different methods and compare them. It's like a bit old, 2019, but it's still very good in comparing how to think about comparisons between different methods here. So there they used 16 classifiers and they categorized them based on their machine learning, the basic machine learning method that was behind them. For example, SCPredict is SVM, Single R is a correlation-based, SCVI is a neural network, for example. I mean, these are old. Now we have SCANVI as well. Like, I mean, SCVI is like an autoencoder that you have, but a variational autoencoder. And we have the new version of it as SCANVI that is more specific for cell annotation. And that is also autoencoder, but uses another neural networks inside it to annotate the cells. And then they also added 6 off-the-shelf classifiers. This means that they use all these classifiers that they were trained based on a certain data set. They were like tuned for a certain cell atlas or reference data that they were using when they were introducing this method. But they, since they wanted to see if, for example, if the general classifier that is coming from like scikit-learn without knowing anything about single cell data can work really, can generalize here when we are using different reference data or not. So they wanted to compare that one to see how much those classifiers are tuned for certain data types or certain situation. They compared those as well. They used the k-nearest neighbor, for example, SVM, and then they also added this SVM rejection with this rejection functionality. And they divided the whole paper or the test that they had done in 3 different experiments. Experiment 1 is intra-dataset evaluation, which is not a realistic evaluation, but it's the fair way to compare different methods, because we don't want the variation between the data sets also to affect the output here. So they wanted to keep that one very fair. So they should perform reasonably similar if they are using the same data set. So what they did, they do 5-fold cross-validations, meaning that they take the data set, divide it to 5, they get 1/5 of it as a keep out, and then train it with the other 4 group, and then take the last one as a test and repeated that one. And they also evaluate the performance using F1 score, that is a summary of precision and recall. And also they look at the unlabeled cells. And this F1 score is ranged between 0 to 1. It's the first, 1 is the best score and 0 is the lowest. So the first thing that they did, they used pancreas dataset, which is of course one of the datasets that's like very easy to classify. The cell types are very distinct from each other. Because they are producing hormones, you have very good markers for each cell type. So they use it as like easy data set to compare these methods to see how they perform and expect, and they were expected to perform very well. So these are the 5 data set that they use, and also for CellBench, also it's like a cell line that they cell line is of 3 different cell lines, and I mean most of them are performing well here. I mean they are all of them, of course, performing very well. It's just like performance 1, and they could also see that they this like SVM rejection, they are doing reasonably well or outperforming the rest here. Then they used brain dataset, and they saw that for example, they have these 3 levels of annotation. They have brain with 3 category, 3 cell types, and then 16, and then 92. When they have this task of like annotating cell type annotation based on only these 3, all this classifiers perform perfectly. When just the cell type, the complexity increases, like the number of cell types increases, then they start to like perform less compared to like when they only have 3 cell types. And then they added when it increased, like the number of cell types, then the performance declines as well. But one of the things that to keep in mind is that performance is not the only thing that, like F1 score is not the only thing that you need to look at. For example, for SVM rejection here, we will see that it has 0.98. It's almost 98% are labeled correctly. But then we also see that 20% are unlabeled. So we need to have like look at the F1 score and unlabeled ones. Because when for example, in a situation that they will do 100% unlabeled, the F1 score is 1. So we don't want to have that one. We want to have higher F1 score with low unlabeled one. Because just remember, this is intra-data set annotation. So we already know what are there, and we are using the same data set for test as well. So we expect very low unlabeled cells there. And then they also did this test by for PBMC data, which has, of course, more it's more challenging task to do. So like cell types are very close to each other. There are overlaps between cell types. And here also they included those methods that they couldn't include, like those gray ones here. It's because they needed prior knowledge. They needed the markers. And since they didn't have it, then they weren't really included in the analysis. But here, these data sets, 2 data sets, the first one has been sorted immune cells, and this is the whole population of PBMC. They had the marker genes with the data that they used, but as you see, the asterisks here, they included only like 3-4 cell type markers for 3-4 cell types, not the all cell types. We see that they perform well, but just imagine that they are only performing on those cell types that they had the marker genes for. And also, this is like the first 5 here that we will see, they are doing very well, SVM, and SCPredict is also SVM based, and SVM rejection. They are performing reasonably well, but SVM, I think, perform better here. SVM rejection has like more rejection rate, especially when you have like more cell types here. So if we just put all of these ones that we look at together, and we will see the top these top 5-6 ones, which are mostly off the shelf, like SVM rejection and SVM here. They outperform the single cell based classifiers. And also we also understood here that the complexity of the data is also important here. So algorithms perform differently when the complexity changes. And the complexity is not only based on the number of cell types, it's number of cell types and also how far away the cell types are from each other. Like a data set that has maybe 80 cell types, but they are very distinct, might be has lower complexity than a cell type that has like 6-7, but they have so many overlaps between them. So that should also be considered here. Well, for the experiment 2, they did inter-data set evaluation. And they trained on one data set, evaluate on the another. So it's more realistic. For this, they used this data set that they had 2 replicates of PBMC, and they used 7 different single cell RNA-seq platforms. So they use this data. For example, they trained on one method and tested on the rest, and they repeat that one for all 7. And they also could do, for example, train on one replicate in a specific protocol and then test on the second replicate. So this here, they expect it to perform well because the method is similar and also they are just replicates. For example, here the rows are the classifiers, and the column you will see the protocol that has been trained on, and then these down here are those that have been tested for. For example, here the first column on the left has been trained on 10x version 2 and has been tested on SmartSeq 2 and for the rest. And then they also add, for example, that replicate 1 from 10x2 as a training and then replicate 2 as a test. And they did this for all the 7 protocols. As we see, like here, for those ones that have been the replicates, they all perform reasonably well, except for like CellSeq 2 here. And we see that they are like these well-based methods, that they are not very replicable, I think, as I remember. And also on the right one, right side, you will see the the performance per classifier. And just here we are getting like SVM rejection that was performing the top one on when we were looking at inter dataset evaluation, but now it's not performing very well. But when we look at that, we see these blue ones are mostly when it's dealing with well-based methods, and then if it's either trained on 10x or like a droplet-based method and then test on SmartSeq, or just like tested, trained on SmartSeq and then tested with the other method. So it seems that it's not performing very well there. And then here also it's performance based on the tests here that we see. Of course, these trainings are the performance when you're using the same protocol with different replicates is high, except for CellSeq 2. So I can pass this one, and also this one, all the methods that have been trained on SmartSeq 2, they didn't really perform well on different trainings with different tests with different methods. So the experiment 3 that they did was just a rejection evaluation. So they wanted to see how well they can reject. What they did here, they did train, for example, on they chose 2 different, very distinct data sets and trained on one and tested on the other one. Here they, for example, trained on PBMC and then tested on pancreas. What they expected to be like 100% rejection or like close to 100% rejection, but they realized that we will see here that not all the classifiers are doing that. And also they did it with different conditions for human and mouse, and still they are not performing well. To make it like more compare it in a more like realistic or scenario that we sometimes have in the data sets when we are want to do cell annotation, they left out, for example, T cells. Like sometimes the classifier is trained on the standard data sets, and then we have our own data sets that is lacking or doesn't include a certain cell type. So we want that one not to be mislabeled with a different cluster. We want it to be rejected as unknown, so we can take care of that outside of the classifier. Here they took out the T cells from the training data and then tested on it, and they still saw that they are not really performing well. They are not rejecting them, and that's because T cells are also, of course, they are overlaps and they are similar to another cell type probably in the data set, so they are getting that label instead. And that also varies in each data set, in each method. And that's because they are using a different threshold, different way, different decisions that they are making inside these classifiers when and where to reject a cell. To see if they can also fine tune that when they went even like in subgroups, to see if that will help to get higher rejection, but that wasn't the case here. So now we know that the rejection is very difficult for the classifiers to do. And then they put everything together, and at the end they also mentioned that some of these were off the shelf classifiers. Those are that like generally they haven't seen any single cell data. Sometimes they perform better, so you don't need something very complicated to do that. So conclusions so far is that simple off-the-shelf classifiers outperformed dedicated single-cell methods, and prior knowledge and markers they are not useful sometimes. They don't improve the performance, and rejection is also difficult. And they also have this pipeline that you can really run it on your data and see how it works on your data. There was another benchmarking paper in 2021 that was also comparing different methods, but here the number of the methods that they were comparing was more, and they were separating the methods based on these classifiers, what call it, the single cell annotation methods based on the machine learning method that they are using. They call it, some of them, eager learning. Those that were classifiers, they were using, for example, SVM, random forest, or neural networks. And those that were dependent on nearest neighbors, they call them lazy learning. And those that were depend on scoring function as market learning. I didn't go into the details here, so we have so many slides, I removed them. But then what they did here is that they used the mouse dataset, as I remember it, and they compared like the other paper, like interdata sets and intradata set across validation and everything, that they were that five-fold cross validation that they were doing in the other paper as well. And then they compared them based on F1 score and accuracy and on labeling rate. And then here in this paper, they also used a tumor. They tried to use it on the tumor data set, because I know like annotating cancer data sets is more challenging than the others, because there are a lot of atlases on normal samples, but not so many on the cancer samples. Then they also use these long data sets for cancer, for tumor cells, to see which one of them are performing well, and then they categorize them and they order them based on the performance per category. What they realized here also, for example, here was that classify R was performing, was one of the top one, as we predict, was performing reasonably well. But classify R is one of those correlation-based methods. That is just, for example, doing better with the tumor cells, because then it doesn't really make a decision, it just correlated, and then it has like rejection option as well. And also some of these marker-based methods and lazy learnings are also doing well for cancer cells. So there are a lot of details in their paper. You can look at that if you're interested in. So now we know the challenges are like about these methods. They vary based on the data set, which data set they have been trained on. That can also play a role. The train and test, how close they are, like are they trained on SmartSeq or they are trained on 10x, so how close they are. So we have a lot of uncertainties still, like to that point, but there are more recent methods that they came. This is a CellTypist that you use in the lab, I think, the Python lab. Here, what they did is that it's also for immune cells across different organs. So they collected data from 20 different tissues from 19 data sets. And what they did here, the method is, the classifier is based on logistic regression with a stochastic gradient descent to minimize the cost function here. So what they did here in the described in the paper, they combined and harmonized these data sets. They scaled the data based on what they wanted, and also did feature selection before the training. So what they did is just like first they used the whole genes and then run initial training, and then they chose 300 genes per cell type and then they combined that and used those features for the main training. And they also divided the data sets to mini batches to speed up the process because the data set was huge. So they didn't want to spend so much time on that one. And when we have an unannotated data, we do transform the data, normalize it based on what they need, and then run it through the model. We get annotation per cell. And then they use very high resolution clustering and then majority voting per group to assign a label per cell population. And then they benchmarked it with other label transferring methods that they thought that they are using, like label transferring method, like similar to what they are doing here. And they also showed that like CellTypist and logistic regression, the off-the-shelf one that hasn't been trained by single cell before, can perform very well. Like they have very high precision, and their F1 score is also high. Of course, they are just comparing it with what it's similar. It would be really interesting to see those with the other methods as well, not only those that are using the similar machine learning method. So the other thing is, of course, like generative learning that came with this ChatGPT era. So they try to use this generative pre-trained transformers for that. Nikolai talked about this in the integration slide present lecture, because this is not only doing annotation, it does a whole bunch of like analysis. So this is run on you can do almost all the downstream analysis using those. It depends on the integration that it does, and then when you get the output from it, you use the latent space for your downstream analysis. What it does for cell type annotation is that it will get, so first you pre-train it, and then you fine-tune it. And one of the tasks for fine-tuning, for example, can be cell annotation. Then you train it with the data set that you have and use the output from a transformer from SCGPT. Give it to a like neural network, an extra neural network there to assign the label. So you add another layer of classifications there, and you will assign the cell type annotations. This used a huge number of, it's like 33 million cells across different tissues. They have very huge data set there. I haven't done it myself, so I don't know how it works. In their paper, they say that it performs very well when they compare it with, of course, with the similar methods. They do better than the older versions. The other one is SCANV, and that Nikolai also touched upon in the other lecture. So they are based on the integration. So you harmonize, you get the new data, you will integrate it with the data, and then you will want to assign the cell types, like transfer the labels. Here also what it does, it's just exactly similar to SCVI, But then what it does is just like it uses the latent representation of the data and get the information about the position of cells and add a classifier at the end of that. So it's a classifier under this variational autoencoder. Then using that classifier can annotate the cell types for you. The other effort that I had seen that there was a paper on, so people try to use ChatGPT for cell type annotation. So we know that the traditional way was that you need like a human expert that he will sit and do annotation based on the markers. But what they did here is that they use ChatGPT instead. So it's just like a command and different commands that they use. For example, for different diseases, these are the markers that I get, how which cells they are associated to, and then yeah, it's like a chain of commands, and they called it GPT cell type there. And they also showed that it performs better than singular and other methods. So yeah, instead of like an expert reading papers and finding the markers, ChatGPT is doing that for them. So that's also one way of doing that. So the challenges of annotating the cell types in the data is that there is no ground truth. So in all these benchmarking that we looked at, they use the annotation from the original data set as a ground truth. So then they want to see if their performance is high and low, they compare it with that one. Of course, in some of these data sets, they were sorted, so they know, or they were separated cell types, so they know exactly what they are. And the cell type the annotation by the authors of those papers are not included in this analysis, but for the other ones, they were really dependent on how accurate the initial annotation was. And the other one is like biology is complex, cell states vary continuously, so we have different cell states. You can't probably, like most of the classifiers can't really define, is it a different cell state or is it a different cell types? Should they reject or not? So they are not trained for that. Separating these cell types, as I said, is imprecise, so you can't really draw the line there. Cancer cell annotation is even more challenging because it's very heterogeneous. They have different clonal expansion, they have different, different profile of gene expression in the same kind of cancer cell population. And also it's very crucial that annotation methods highlight the uncertainty. So then we know where to go, when and where for which group of cell population we need to do manual annotation. So is there any way that we can benefit from these automated cell type annotation that minimize all these issues? That is a question here. There has been some efforts to tackle this issue that we can still do automatic annotation, but we would get the best performance out of it. So we want to get the rejection as well. So we don't want to have be only dependent on one specific method here. So one solution here that in this paper they presented is you do use different methods and then use popular vote or a voting system to decide if how certain you are on that cell type. In those cell types that not all the methods agree on, then you will get a higher uncertainty there. So you can go and look at those cell types and do manual inspection there if you want to. And in their paper, it is a very nice paper. I will recommend reading that. Yes.
Just a question. Do you know if all of these methods are using the same reference, or is there also a difference in what references are being used for all the different methods?
I think as I remember it, they use them because they have different options. You can train them from the beginning, so you get the architecture or everything for them, and then you train it from the beginning. You have three options, I think, in their pipeline. You do train them based on completely new thing, so they are not pre-trained, uh, as I remember it. Um, I think they tried to fix that one, and they used a certain data set that most of these, for example, CellTypist was um,
The
ran on.
CellTypist this might be pre-trained. No, I'm just thinking that it's really like with different references and still getting consensus would be the strongest sort of evidence, right?
Yeah. I can't really say exactly.
I will have a look myself.
Yeah, I don't know. I think I don't remember it. But I know they had some concerns there, and they described it in their supplementary, that they did some stuff there to make it like quicker. If you want like the training to happen, it's going to take, for example, I don't know, 1 hour, 2 hours. If you just use the train that they did themselves, then it takes less. But I assume they did train them based on certain data. I don't know. So, anyways, they used 8 methods here. And then what they do is just they get votes, for example, from like to 8. If all the 8 methods that are there, the classifiers agree on, then you get them have a consensus score there that they are like we are very certain that is this cell type. Um, the other paper that also have been thinking similar to that is this, and that was on cancer data set. What they did here is that they didn't do something extensive like that. What they did is just for cancer cells, you have two different methods, like copy number variation based methods and reference based methods. So that is, and then they thought they claim that these two are the state of the art in the field for cancer data annotation. So they chose these two methods there. And then what they do here is that they say that reference-based methods that have been trained on cancer cells can perform well, but not on every case. They have what they do is that this method at least is using the reference, and then it's very good at annotating the normal cell types, but all the epithelial cells are annotated as tumor cells. Um, what they did in this paper, they tried to compare it with the output, for example, for copy number variation based methods. And then there, they dividing the tumor cell, the data set based on normal and tumor. So they try to combine these two methods together to define a different method that is using, for example, using the reference-based method to separate epithelials from the rest. And then now we have what are the epithelial cells there that should be normal and tumor cells. Then they run CNV-based method on only epithelials with a group of the other normal cells as a reference. And then they get the output here that they have the normal cells that were coming from reference-based methods. They have epithelial ones that all were labeled a tumor, and then run it through CNV-based methods, and then they get normal epithelial and tumor cells. And they also compared it with CNV patterns and everything, and they claim that this is doing better when they are comparing with different data sets. So maybe that is one way to solve the issue for cancer data. We don't know yet. We need to really evaluated ourselves. So, but it would be good to keep in mind that only using reference-based methods and grouping them based on like most of these methods, they are not performing very well on cancer data sets. So just keep in mind that one. And here also they had then this example here for pancreatic cells. When they use reference-based method, they get these normal and tumor cells that in the original data they are labeled as ductal cell type 1, ductal cell type 2. And reference-based method are just grouping all of them as tumor. CNV-based method grouping only like the one which is, I guess, ductal cell type 1 as normal and the rest as tumor. But when they do consensus, then they can separate these epithelials between tumor and normal epithelial based on CNV. And for breast, for example, data set, it's one of the data sets that they don't have very strong copy number variation. Just keep in mind that copy number variation is not also something that all the cancer data sets has. Some cancers are stronger on copy number variation and some are weaker. And these methods for example, CNV-based methods, they are forcing a clustering at the end of the process to divide them into two groups of normal and tumor. And if, for example, in this case for breast that we have very weak CNV, by forcing them to divide into two groups, it just labeled a lot of normal, like immune cells even as tumor here. So it just gives you really a lot of false positives here. But by using like reference-based methods, also you can really fix that one. So you understand that, okay, that's not the case. And that's because only the CNV signal is low. So you know that one is also very dependent on this cancer cell type. In the lower down here, you will see which ones, for example, CNV-based methods worked bad really, for example, for breast, for head and neck and lung and kidney, CNV-based methods are not performing well. And for reference-based methods also doesn't perform very well with pancreas samples. But then when you combine all the positive parts of these two methods, the consensus ones perform better here, at least for those specific cases.
So the CNV is inferred from the same single cell dataset as the reference base?
Yes, yes. So to summarize, we say that cell identification is moving from unsupervised to supervised learning. You should always check what reference you are using. If you are using like a reference data set that you have, you manually annotate it, and then now you want to use that one or not. The more similar the references to your data, the better the prediction. If they have been trained and tested on the same technology, or they have been trained on the same technology as you are using your testing data, it's very important. And also how much you trust the self-type annotation that they have done. Some of these consensus methods help you, even if there is a miss annotation in the reference data sets, you can also capture those. For example, in the paper like PopV, they really found some of these miss annotations in the reference data, and they claim that when you look at this one and go back to them, you will see some of these issues with the original self-type annotation as well. And also atlases, they don't contain all the tissue cell type and also especially cell states. Look at the differential gene expression and known markers and check that if the prediction gets better. Consensus annotation methods combine the power of other different like those automated methods that based on different techniques. And that can be beneficial when addressing new data sets, new cell types, unknown cell types, or disease samples. You're dealing with those. There are also some general resources that you can look at if you want. And also acknowledgement again, Osa and Ahmed for the slides. Thank you. Okay, yes.
Hi, Fariba. Thank you so much. I have a question regarding the unlabeling rate.
Yeah.
Because many of the times, like in cancer cells, you have like these epithelial to mesenchymal transition states, and you have like many of these cells in these transition states. And do they like even, is it possible like when you take a reference data set and it has marked with this is like the transition state markers, what is the chance that like those transition states are like compatible with your data set, or like how much can I afford to lose on this information?
So if you have the reference data that has been like performing or have been labeled, and the number of cells in that cell population are also high enough. So it's not like only 10 cells that you have labeled that. So you need a bit of variation there as well, the population. Then you can say that most likely you will get that, right? Also correct me if I'm saying it wrong. But if you like don't have like so many cells there, that might not work. But technically, if you have it as a reference and use your own reference for the method, that should do that annotation for you, right? Because especially the method what they're using, because some of them they are just using depends on the method. Like then you project your data that you are testing on the other data set, you integrate that first in some of the methods, and then you transfer the label. So technically, if any of your cells from the new data set is grouping with those cell types, there is most likely getting that label, right? So it depends on the algorithm that you are using as well.
And in this case, I can use different algorithms to
Yes.
test like
Yes,
how
those that do label transferring, yes.
Okay. And regarding the techniques that you said, like when you were showing this slide, like sometimes when you have SmartSeq and like 10x, like there is not we have to test like different techniques to see like if the labels are transferred, or do you suggest like if I have a 10x data, I should always try to see like if there is a 10x data reference available and use that?
I think so, yeah. I would do that.
Can I just comment there? Because I would say, let's say that you are working with like a disease state or a tissue that is rare to find. It's still better if your reference is SmartSeq, but very close biologically to your data set.
Okay.
Than to have something that is like only healthy controls from 10x. So that also, I think the biology matters more than the technology still, just to keep in mind.
Yeah, that's true. Yeah, thank you.